Hey guys, how you doing? I hope you guys all enjoyed your break and you're all staying safe and sane. So let's just get into this. I don't want this video to be very long. Now, today we're going to be talking about gene regulation and what does gene regulation do? Basically, it turns off transcription when it's not needed. And why would you want to turn transcription off? Because you don't want to make every single type of protein that DNA could code for, because number one, you'll be there all day long. And number two, you're going to be wasting your resources. You're going to be wasting your energy, and they're just not needed. Now, gene regulation is going to allow us to transcribe genes for proteins that are needed. Now, for example, what you're looking at here is going to be the lac operon, okay? Not the RNA polymerase, not the active repressor, just this whole segment here, okay? It's going to start with a regulatory gene, then we're going to go into the promoter region, we're going to talk about the operator, and then at the end, we're going to talk about each of the individual genes. Now, an operon is going to be a set of genes that are regulated together. For example, the LAC-Z, the LAC-Y, and the LAC-A gene are all going to be regulated at the same time. Now, when it's regulated, what does that mean? It means that something is going to determine whether these genes are going to be expressed or not. Now, a segment of DNA is going to transcribe into a message of mRNA in the nucleus, and then it's going to fit out of those nuclear pores, and that message is going to go into the cytoplasm. It's going to find the ribosome, and that message will be translated into proteins, or in this case, enzymes. Now, with gene regulation, you're always going to have an operon. And again, an operon is going to be a set of genes that's going to be regulated together to determine whether or not transcription is needed or if it's going to happen at all. Now, for this example, we're going to be using prokaryotic DNA from bacteria and E. coli, but we'll go into that in a little bit. But it's just easier to look at prokaryotic DNA than it is to look at something that's more complex, like a multicellular eukaryotic organism like us. Now, okay, you're going to have a regulator gene, okay? And what this regulator gene does is it codes for the active repressor, okay? And what this active repressor does is it binds to a spot where it fits perfectly into the operator. Now, normally, when this active repressor isn't there, okay, the RNA polymerase would bind to the promoter, and then it would run the segment of this DNA, and it would make an mRNA message that would eventually be translated into proteins. But this is an induced operon, which means that it only turns on when it's needed. Now, in this case, there is no lactose present, okay? And lactose is the sugar from milk. It is a disaccharide. Its monomers are going to be glucose and galactose. Now, so in this example, as of right now, there is no lactose present. Therefore, this active repressor is going to fit in perfectly right here on the operator. And what does that do essentially? Essentially, that blocks the RNA polymerase from running its course down the segment of DNA and making that message. So it's kind of blocked. All right. But once that there is some of this lactose here, and I want you guys to imagine all these green circles as lactose. Now, this is an induced operon, which means that in the presence of lactose, okay, there's going to be a little spot in the active repressor. And once a molecule of lactose binds to that spot in the active repressor, it then inactivates the repressor. Therefore, the repressor falls off of the segment of DNA. And now we have something called an inactive repressor. And what does that mean? That just means that when the lactose bound to it, it changed the conformational shape of the repressor. Okay, now this repressor does not fit perfectly like it did before onto the operator, and it falls off of the segment. Now the RNA polymerase is allowed to start at the promoter and move its way down the segment of DNA, making its mRNA message. And if RNA is not able to make its way down the line, mRNA will not be made. So after okay, so in milk there is a sugar called lactose, and we have a type of bacteria in our intestine called E. coli, and our relationship with E. coli is going to be a mutualistic symbiose. Okay, what does that mean? That means that it's a win-win for both us and the bacteria inside of our intestine. These bacteria help us digest certain types of foods, and in return, the bacteria get nutrients from the foods. So it's a win-win. The E. coli in our intestines will take the disaccharide lactose and break it down into its two monosaccharides, glucose and galactose. Just remember that di means two. Okay, like a bicycle has two wheels. But in this case, it's di, not bi. And this is just another prefix for two. And saccharide means sugar. So if it's a disaccharide, that means that it's made of two monomers. And in this case, it's glucose and galactose. So in order for your cell to be able to use and break down this this lactose right here, it needs to be broken down into its component parts. And again, transcription is only going to happen if this lactose is present in the environment. If there is no lactose in the environment, okay, let me just move these over here. Okay, this active repressor fits, and RNA polymerase is now blocked from moving its way down the line. Therefore, it can't make its mRNA message. So now that we have some lactose in the environment, the lactose binds to the active repressor. And when it does bind to the active repressor, it changes its conformational shape. Therefore, it does not fit in perfectly into the operator. Okay, so let's just move this over here. Okay. Once the RNA polymerase is able to make its mRNA message, okay, and these are going to be supplemented with nucleotides, and there's actually going to be, a code. This is how you guys have practiced transcription and translation. Now we're just putting it into gear. So now remember, this is an induced operon. It only works if there is lactose present. Okay, no lactose present, no mRNA. No mRNA, no lactase enzymes. Okay, so let's move this here. So, sorry. Okay, so when after the mRNA message is made, the RNA is able to run the track. Said that stuff. Okay, so good. So once the RNA polymerase gets to the LAC-Z gene, okay, it is going to be transcribed into an mRNA message. And once it's transcribed into an mRNA message, it's then going to be translated into lactase enzymes. Now, the LAC-Y gene is going to code for something called beta-galactosidase. And what beta-galactosidase does is it breaks down. I told you I want you to think of this as a disaccharide with two pieces. I just did it for simplicity because it would fit into this little section over here. But in reality, this is going to be two monomers put together. And it's going to be lactose. And what this very first enzyme that is made from the LAC-Z gene is called beta-galactoside, and it cleaves the lactose into its two monomers, into its two halves. The lac Y gene is going to code for beta galactoside permease. And what permeases do is they allow more of that type of material into the cell. So in this case, we have beta galactoside permease, means it's going to let more beta galactosides into the cell, aka sugars, aka lactose in this case. And finally, this beta galactoside permease is represented by a diamond, and the beta galactoside is represented by this hexagon here. And the very last gene that is on the lac operon is going to code for an enzyme called beta galactoside transacylase. And yes, it's a very long name. You do not need to know the names of these enzymes, but what this enzyme does is it moves an acyl group from acetyl-CoA to the beta galactosides, aka the sugars, aka the lactose in this case. So I'm trying not to make this video very long. If you guys have questions, please put them on the discussion board posted with the assignment. Real quick before I end the video, I'm going to run through this again quickly. All right, so let's put this back over here. Put these guys down here. Change the color. All right, let's move our RNA polymerase back. Okay, so from the beginning, all right, there is going to be a section of the promoter called a regulator gene. Now, this regulator gene, what it codes for is this active repressor. And in the present, now this is an inducible operon, meaning that it only turns on when lactose is present. So in this case, okay, there's no little green circles, there's no lactose present. Okay, and this active repressor is going to fit perfectly into the operator. Okay, and what that does essentially is it blocks RNA polymerase from moving down the line, creating its mRNA message. Once we introduce a couple molecules of lactose, lactose molecule will bind to the active repressor. And essentially what it does is it creates an inactive repressor. And you could see that the shape is different. Now, this inactive repressor does not fit perfectly into the operator, therefore it falls off. Now the RNA polymerase is able to start at the promoter, run the length of the DNA to make its mRNA message. I'll leave this here. Once we have our mRNA message, it is then going to be translated into proteins. Now we just put 3 shapes here, but the lac-Z gene is going to code for beta galactoside, which cleaves lactose into 2 monomers. Then the lac-Y gene is going to code for something called beta galactoside permease. And what a permease does is it just allows more of that material into the cell. And then we're going to have the lac-A gene, okay, which is going to code for beta galactoside transacylase. And that just moves an acyl group around from acetyl-CoA. So in the end, lac-Z, okay, codes for our hexagon, our galactosidase. Lac-Y, our permease, and lac-A is going to be a transacylase. You guys do not need to know the name of these enzymes. You just have to know that they're different lactase enzymes. Okay. That being said, the rest of your assignment is, well, not the rest of your assignment. Another part of your assignment is going to be an ed puzzle on the amoeba sisters. Hopefully that it sinks in a little bit better when you hear it from someone else, as well as myself. I miss you guys terribly. I hope you are all well. I hope you are all sane, because I'm going crazy here. Have a good day, guys. Bye-bye.